Hello, my name is Elina Stamatelatou, and as Claudia introduced me, I am an application engineer at BIOEMTECH. In this talk, I will take you through an overview of preclinical metabolic PET and present some case studies that show how the β-eye system can support different research applications. BIOEMTECH is a Greek company based in Athens with a team of about 55 people coming from a wide range of disciplines: physicists, engineers, radiochemists, biologists, and veterinarians. Our work focuses on two main activities. The first one is that we design and develop small animal PET, SPECT, and optical imaging systems, our series of instruments that we call the eyes. And secondly, we provide complete preclinical imaging services for pharmaceutical companies and research groups who need to evaluate the behavior and effectiveness of new compounds. Radiopharmaceutical development is a long and selective process, and only a very small fraction of candidate biomolecules eventually becomes an approved tracer or drug. Before reaching clinical research, there is an extensive preclinical phase made up of several distinct steps, from in vitro testing to radiochemistry validation to animal model preparation and, of course, in vivo imaging, which is where we are most active. Our role is mainly in step 4, in vivo imaging and quantification. This is the stage where we evaluate biodistribution, pharmacokinetics, target engagement, and metabolic behavior in living systems, providing essential data that guides the rest of the preclinical pipeline. Small animal imaging is a powerful tool because it allows us to visualize biological processes non-invasively inside the living organism. A major advantage is the ability to follow the same animal longitudinally, capturing dynamic changes in tracer distribution, disease progression, or treatment response, instead of relying on a single endpoint. Importantly, in vivo imaging is not just qualitative. It provides quantitative biodistribution, pharmacokinetic, and mechanistic information, data that are essential for validating disease models and for early evaluation of new diagnostic tracers or therapeutic agents. This level of insight cannot be obtained from ex vivo biodistribution alone. Another key benefit is the reduction of animal numbers. By imaging the same animal across multiple time points, we get biodistribution, kinetics, target engagement, and biological dynamics in a single experiment, fast with fewer animals and fully aligned with the 3 R's principles: replacement, reduction, and refinement. With in vivo imaging, we can answer essential biological questions that guide the entire preclinical process. First, we can determine whether the biomolecule actually reaches the target, how long it remains there, and when it reaches its highest concentration. Second, we can characterize its biodistribution and stability, where else it goes in the body, how long it circulates in the blood, and how stable is it after the injection. Thirdly, imaging helps us to optimize the full experimental protocol. What happens in the very first minutes, which injection route and dose works the best, and what the ideal time points are for biodistribution or tomographic imaging. So overall, imaging provides a complete dynamic understanding of tracer behavior in real time, something that we cannot achieve with endpoint methods alone. One of the most powerful applications of small animal imaging is metabolic imaging. Metabolic imaging focuses on the biochemical activity of tissues, how cells take up glucose, use amino acids, synthesize lipids, or manage oxidized stress. These functional changes often appear very early in disease, long before any anatomical differences becomes visible. And this is exactly where PET excels. PET is extremely sensitive to follow metabolic changes and allows us to quantify them directly in vivo. It also gives us access to a wide family of radiotracers that each target a specific metabolic pathway, so we can choose the biology we want to study. Another major advantage is translation. The same PET tracers and the same imaging principles can be used in small animals and in humans, giving us a direct bridge from preclinical research to clinical application. For all these reasons, PET has become the reference method for metabolic imaging, offering sensitivity, quantification, pathway specificity, and translational power. Each isotope provides a unique combination of half-life, positron energy, and radiochemical compatibility. And this flexibility allows us to tailor the tracer to the biological process and time scale we want to study. In this slide, you can see a summary of metabolic PET tracers that are frequently being used. For instance, FDG, which is the main tracer for glucose metabolism. It covers tumors, inflammation, and it is very important in neuroscience for showing hypometabolism in Alzheimer's disease. We also use FDG in cardiology to evaluate myocardial viability after infarction. Beyond producing images, PET gives us true quantitative information, which is one of its greatest strengths. Quantitative PET allows objective comparisons across animals, doses, and treatment groups, something essential for biodistribution studies, therapy evaluation, and pharmacokinetic analysis. Usually, the metrics that that are extracted are percent injected dose or percent injected dose per ml, which tell us how much tracer accumulates in a tissue, and standardized uptake value, which normalizes this to dose and body weight. When we want to understand biology dynamically, time activity curves helps us to follow uptake over time. And finally, with kinetic modeling, we can go deeper and extract rate constants that describe transport, trapping, or metabolic conversion. Before moving to the application data, I should mention that all the experiments were performed using our BETA system. This is our compact preclinical PET scanner. I will not go into full technical specifications today, but briefly, it incorporates state-of-the-art detectors configuration and supports real-time dynamic imaging. The system can also image up to 3 mice simultaneously, which is very, very valuable for comparative studies and for reducing animal numbers. All the results that you will see in the next slides were produced by BETA users. To start with the applications, here is a representative example using fluorine 18 FDG, the benchmark tracer for tumor glucose metabolism. In this study, 2 human colorectal cancer models, SW480 and HT29, were implanted subcutaneously in mice. Each group consisted of 10 animals. FDG was injected intravenously and imaging was performed 60 minutes post-injection with a 20-minute acquisition time. The images were then analyzed using Imalytics, where the SUV mean and SUV max values were calculated for the tumors and for reference tissues to compare metabolic activity between the two models. The aim was to assess how aggressively each tumor type metabolizes glucose and to quantify differences in metabolic phenotype. Qualitatively, when we look at the images on the right, the difference is very clear. The SW480 tumor lights up strongly, while in the case of HT29, a much weaker FDG signal is in the tumor. It is here in the back of the mouse. Quantitatively, in the table, you can see results from those example cases, those 2 mice. The SW480 tumor showed higher FDG uptake with SUV mean of 2.2, indicating enhanced glycolysis, compared to 0.8 in the HC29 case. We also see strong activation of brown antipose tissue in the SW480 case, which is consistent with elevated systemic metabolism. We will discuss this point in the next slide, though. Brain and liver showed the expected physiological and comparable to the 2 cases uptake levels. All these metabolic profiles highlight intrinsic biological differences between tumor lines and show how PET quantification supports precise model selection and therapeutic valuation. PET can also reveal unexpected biological issues, and that may affect the study. In this case, while imaging the HC29 mouse of this example, a strong FDG signal was noticed in one of the hind limbs here. This signal was not related to the tumor, but was instead consistent to inflammation or infection. Such findings can influence biodistribution and dosimetry calculations, and acting on them improves reliability of our data. In this example, during the FDG imaging of the SW480 mouse, a strong activation of brown adipose tissue was observed. Brown adipose tissue is highly metabolically active, and when activated, it takes up significant amounts of FDG. This is a physiological response, which is triggered by temperature, stress, or metabolic state. Identifying this kind of patterns early ensures that we interpret biodistribution results correctly and maintain experimental quality. In the next application example, you see preliminary cardiac imaging results from a mouse model of myocardial infarction. On the left, you can see the ECG tracers, how the expected differences, where we can see the expected differences between a control mouse and an ischemic mouse. To evaluate the metabolic consequences of this injury, FDG cardiac PET was performed. In healthy mice, FDG uptake across the myocardium is more uniform and symmetric, while in the infarcted hearts, we show reduced or heterogeneous uptake, indicating impaired glucose metabolism in the affected regions. These results show how ECG and cardiac PET complement each other, and allowing us to non-invasively assess both the functional and the metabolic impact on myocardial infarction, even in small animal models, like the mouse. A different application, in ovo PET imaging. This uses the chick embryo, which is a highly perfused and metabolically active system, making it ideal for rapid screening of new radiotracers. Here, H460 tumors that overexpress the XCT transporter were imaged using fluorine 18 FSPG. Specifically, 2 megabecquerel were injected, and a 10-minute acquisition was performed 1 hour post-injection. On the right panel, you can see the optical view for orientation, and then sagittal, coronal, and axial PET slices. The tracer shows clear tumor uptake here, while by distribution in the embryo, we can see clearly the liver, the kidneys of the embryo. The main advantage of this model is that it acts as an early decision-making step. It allows us to evaluate targeting, contrast, and basic pharmacokinetics before moving into full mouse studies, saving time, cost, and animals, perfectly aligned with the 3R principles. In this final application example, we can see how PET can support early-stage drug research. In this example, 3 mice were imaged simultaneously at a 6-minute scan, 12-minute scan, and 18-minute scan. This gives biodistribution information, and instead of 3 separate biodistributions that would each require animal termination, dissection, organ isolation, and gamma counting, we obtain quantitative whole-body PET data. This is faster, as we can get information in a few minutes, for example, in the first case, 6-minute scans and approximately a 10-minute processing pipeline. Non-invasively, more consistent and much more informative, while greatly reducing experimental workload and the number of animals required. To conclude, preclinical molecular imaging provides us with a powerful quantitative and completely non-invasive way to study tracer kinetics, target engagement, and biodistribution directly inside the living organism. PET remains the most sensitive metabolic imaging modality, allowing absolute quantification of short-lived radiotracers and giving us access to fast biochemical and metabolic processes that other techniques cannot visualize. What really enables progress is the integration of chemistry, physics, biology, and imaging technology, creating an end-to-end workflow from radiotracer development to in vivo validation and translational application. Finally, you've seen examples today ranging from tumor metabolism, myocardial infarction, inflammation, and early-stage drug screening, all the way to in vivo imaging. Together, these highlight how PET accelerates discovery while reducing animal use and improving biological insight. Thank you very much for your attention.